Next, we have Eunsu from the Boltz team. He's at MIT.
Okay, hello. I'm Eunsu Hur, studying mathematics at MIT, advised by Professor Wei Zhang, Tommy Akola, and Jonathan Weissman. And I'm excited to present BoltzGen, a general binder design model that a huge team developed, and I'm happy to speak on behalf of this great team. And our model is heavily structure-based, opposed to the previous speaker, and I think we should have a debate sometime. So to motivate the need of such a binder design, I want to start with an example of the TERT-telomerase. So this protein here is identified by our collaborators who's working on glioblastoma, and TERT-telomerase is used in the cancer cells to repair DNA. And the collaborators wanted us to design a peptide that binds to the pocket where the DNA is usually threaded in and gets repaired, so to block the cancer cells from surviving and cure the disease. So now with BoltzGen, we want to tackle this type of problems, and we want to go in two axes. First, generalization across modalities and different constraints, and also generalization of novel targets. So we wouldn't want our model to be spitting out existing data sets in the PDB and be able to design new binders to novel targets. And to achieve this, we have a couple of requirements, a strong structure reasoning, and the model should understand physics to place atoms in the right places, and have a high expressibility in design constraints. For example, we want to design peptides with specific covalent bonds to some small molecule. So what we want to achieve is, as I said, strong structured reasoning and emulating physics and full controllability over diverse design possibilities. And how we achieve this is, first, we unify design and structure prediction task, which at first can be a bit counterintuitive. And second, we train across diverse modalities, all contexts, and a huge distribution. And finally, we have an expressive design specification language, for example, specifying the binding sites or specifying disordered regions and whether to bind or not bind. So I'll start with structure prediction and design in a unified way. So when BoltzGen designs a binder, it does a couple of things simultaneously. So first, it denoises, is the video? Oh yes, the video is playing. So it denoises all the atoms simultaneously, and the folding task and the design task is done simultaneously. And also, when the denoising happens, the model also reasons how the conformation of the target could change according to where the binder is binding. And finally, the design binder and binder structure and sequence is denoised simultaneously, unlike other models where the sequence is decoded at the end using some inverse folding module. Our model uses a continuous decoding way, which I'm happy to talk about later, but it's something called atom14 encoding, and the sequence is denoised continuously with the structure. And second, for training distribution across various modalities and contexts, we have a diverse set of modalities in the PDB and other public data sets we train on. The reason we do this is because we want to emulate physics in a realistic way. And for doing this, even if we want to, say, design a nanobody binding a certain protein, we should also train the model on general protein-protein interactions so the model can learn how proteins and amino acids interact with each other. And same for other modalities. And finally, for the expressive design specification language, we want to achieve this towards full controllability. For example, say we want to design a peptide, and we want to have a helical structure of the peptide. Moreover, as shown here, we want to place cysteines in the two specific amino acids. Also, we would want to design a helicon sometimes and have some small molecule with covalent bonds bound to the cysteines in the helix. And sometimes we would want to design peptides or proteins with disulfide bonds. Or sometimes we would want to design cyclic peptides with an extra peptide bond between the first and last amino acid. So these type of controllabilities are all encoded in our model. And also, when it comes to binding sites, we can tell the model to say, yes, bind the specific spot over there because it has a certain function we want to inhibit to kill the cancer cells, as I explained for the example of TERT-telomerase. Or we can tell the model not to bind somewhere and rather bind somewhere else. So there's 3 types of binding specifications. And finally, we can also control structure groups, which is basically fixing certain parts of the structure and do partial design. For example, if we want to design a nanobody, then nanobodies have a specific scaffold we usually want to follow. And if we give visibility group 2 and the target visibility group 1, then the model will know that those parts should be conserved and rather reason on the relative conformation of the target and the binder so that it finds a nice place to bind and also decides which interface amino acids will be there. So as a whole, we achieve full controllability through expressive design specification language. So when we give a YAML file to the model, we can have different types of controls, covalent bonds, structure groups, secondary structures, and so with that we can achieve very high specific binders. So this, I explained, is the first part of the BoltzGen pipeline, which is followed by inverse folding, refolding, affinity prediction, analyze, and then filtering. So usually when we do this in practice to generate strong binders, we generate 60,000 designs and go through this pipeline and have a computational set of metrics we use and filter them down to whatever our experimental budget is. So this is an example of denoising trajectories for different modalities, and we can see that the model is finding where to bind on the surface of the target protein. And we get a final structure. So as I said, BoltzGen model achieves unification of design and structure prediction. So we also ran the evaluations for a comparison between BoltzGen and Boltz, a previous model that is a structure prediction model. And we can see that it is very comparable in all modalities to the state-of-the-art models. And unlike structure prediction, we would like to have more experimental validation, not just computational metrics, to see that our design binders actually work well in the lab. So we have over 26 experimental collaborators and expanding on, and this is a subset of them, and they're actually working on real-world impactful problems in biology. And currently we have 8 wet lab campaigns and 26 targets that we have results for, and I'll briefly explain. And this is a summary of the wet lab results, and the ones that don't have a yellow protein bound to it is the ones that we failed, and most of them are pretty successful. And especially for nanobody design, we chose 8 targets which have no known binding proteins in the PDB, and moreover, every single protein that has a known binder has less than 30% sequence similarity, so it's a really novel target. And we achieved nanomolar binding to 67% of the targets, and this here is the number of proteins expressed and the number of the ones that bound, and this shows that BoltzGen can design nanobodies, which is already a hard design task because a lot of things are restricted, and we achieve very high binding affinity, and same for proteins. And one thing I wanted to share additionally was the targeting of disordered regions that we worked with Max Planck Institute for biochemistry for targeting NT1 protein, which is acute myeloid leukemia. And through their assay, we were able to see that our designed peptides were localizing in the right places and shows binding properties. And finally, I'd like to start closing with emphasizing that BoltzGen is fully open source, training, inference code, model weights, everything is publicly available on GitHub. And yes, we expanded the design space across modalities, constraints, novel targets, through unifying folding and design, and expanding through a lot of context and tasks, and with an expressive design specification language, and we have wet lab experimental validations. And however, this is the only first step, and we have many things to do: off-target interaction selectivity, immunogenicity, delivery, degradation, and desired biological effects. And our next step are these, and A lot of them could be potentially related to gene delivery, gene therapies, and yeah, we were happy to collaborate, and I just want to thank this entire team on the efforts, and moreover our experimental collaborators who take all their time and work for this. And thank you so much, and please reach out to our main email, and if you want any collaborations or if you want to see our code paper, GitHub, Slack, use the QR code, and thank you so much.